Good morning. So I'm Diwakar Shukla. I'm part of the plant and silico team here. I'm a professor in chemical and biomolecular engineering, and I'm affiliated with several other departments on the campus. So today I'm going to talk a little bit about molecular models, and Professor Klaus Schulten, who is a giant in the field of molecular modeling, was supposed to give a talk, but he could not. So I will also try to introduce these ideas to you in a very, very simple terms, so that it's accessible. So you all know that this is the plant in silico platform, and so today we are talking mainly about molecular models, and I will try to give some ideas about how can we integrate molecular models with the higher level models, and what is the need of molecular models, and what can we learn from molecular models. Okay, so what I mean by molecular modeling here is simply that we are working in this range of few tens of nanometer at the maximum. So you're looking at individual proteins, you're looking at assembly of proteins. If you think of a photosystem, that's an assembly of it's a large system with a lot of proteins interacting with each other. That would be one example. And we all want to understand these type of systems in atomistic detail, where individual hydrogen bonds that are forming between groups are important for the function. So if you have a charge on a small atom, that is important for function. So you need to represent all these systems in a molecular description to be able to understand something realistic about these function of these proteins in plants. So one way of learning, obviously, about the structure of a protein is to crystallize it, and this is the crystal structure of a protein. But if you think of plant biology, there's very limited amount of effort in structural biology. We don't see that many structures coming out, and therefore computation is one way of bridging that gap where we can go from a single structure to function understanding of the entire ensemble of structures that are available to us. So typically, I'm going to talk about a very specific problem so that everybody understands it here. I will also go into some other type of problems that I work on. So, this is a problem that I've been working on for the last 1 year, roughly. And as you know, the nitrogen use efficiency is really, really low in most of the world, especially if you think of India and China, which are feeding roughly 1/3 of the world. The nitrate uptake efficiency is really low. So, this is one of the projects of the plant in silico where we are looking at how we can improve the nitrate uptake efficiency of plants. So what I'm doing is we are looking at a very, very small, tiny segment of a root, you can imagine, and we are looking at a specific transporter that takes in nitrate from the soil and puts it inside the plant. And how that happens at a molecular level, and what are the different things that are involved, and how can we understand this process. So luckily, let me first just tell you what these things look like. So I thought that there's some such diversity in the audience. This is a small video which explains what are different transporters. It's not that very high quality, but what you're seeing right now are these blue circles, which are transporters, and then everything that you're seeing outside is the extracellular part of the plant. You can think of this as a root surface with transporters and then water that is outside. So just to get you an idea, what is happening is, these transporters are sitting on the surface of the root, and then these small molecules that are floating around in the water, they're all coming into these transporters, and these transporters have either holes in them, which allow specific types of ions to pass through, or if the ligand is really, really big, what would happen is that the transporter has to change its shape to allow it to go in, and these are selective transporters. So now you're seeing it as an open shape, and then it transforms its shape and lets the thing go inside. So how could we simulate this type of a process in a realistic sense with atomistic models? And this is the case with our nitrate transporter, that to transport nitrate, our protein has to completely change its shape. It has to pick up a nitrate, close itself, open it inside towards the cell, and then the nitrates can go in. So this would be a clear picture of it. 3 key states, as they're called: the inward open state, the occluded state, and the outward open state. Obviously, there are a lot of states in between these, which are leading to the transition between these conformations. But what we have so far, there's a couple of papers that came out in Nature last year, a year ago, and we are able to first see just one glimpse of a nitrate transporter in a plant. And this is coming from Ning Zheng's group in University of Washington and Simon Newstead's group in England. And both these papers came out simultaneously, and they both got the inward open structure of this transporter. But we don't know anything about how this is happening except a single picture. And you can imagine this was a great achievement for crystallography, that we are able to crystallize this thing in a single snapshot. But now what I'm going to show you is that we can take this single snapshot and do very large scale simulations and get all the possible conformations of these receptors which have not been crystallized. And eventually, when they will be crystallized, you'll see a paper coming out in a Nature or a Science for an outward open structure of this receptor, but we're giving it to you now with extensive simulations and modeling. One other interesting feature about this particular protein is that there's a specific residue called threonine, which gets phosphorylated, which is really out on the outward. It's not even in the tunnel where the nitrate is coming in. It's really outside, and even then it affects the transport rate by an order of magnitude or more. So you get this distant residue getting phosphorylated. By adding in roughly 4 atoms to this particular residue, you're changing the nitrate uptake rate by 2 orders of magnitude or more. So when this type of thing is happening, how could we explain this at a molecular level, like what is happening? So we know that phosphorylation changes the rate, but we have no hypothesis of why this is doing it. But we can understand this using molecular models. So what I do is, again, this is a very simple stuff. So this is the only structure that's available. It's an inward open structure of this particular thing. We put it in a realistic, atomistically detailed membrane. We put it surrounded with water. We put in nitrate ions around it, and then we see the transport of the ions through these things and how the conformations are changing. Obviously, when you're doing this atomistic description, you're solving the dynamics of these systems using Newton's equations and some forces that are being applied to it based on purely physics-based model. So there's no experimental input here. It's all physics-based model. It applies to this protein and to any other protein that we can think of. So we are going from here, and even for water, we have an atomistic description of a water. So you can think of this system containing 100,000 different entities from which you have to calculate forces every femtosecond. And these processes are happening on a millisecond time scale. So you can now do a jump from 10 raised to the minus 6 to 10 raised to the minus 15, and we are talking about a billion time steps that need to be solved for 100,000 atoms. So it's an enormous task to do these things, and that's why when you're going to look at any of the molecular modeling manuscripts or papers, you're going to find people running simulations which are a nanosecond long, right? A nanosecond itself is a million times, but we want to make that jump from a million times to a billion times of calculations. And I'll show you how we have done that. So again, this is the problem. Obviously, this is a famous statement from Paul Dirac that everything that we need to know about chemistry and physics is known. We just need to solve the equations, and that's true even now. So roughly 100 years from now, from when he said it. So we are at this atomistic level, and what I'm showing you amount of physical detail. So if you go to a very, very highly detailed model where you are taking into account the distribution of all the electrons, you can only do these type of calculations for like 2 atoms, a single water molecule. That's it. We cannot go beyond that if you're doing a correlated method like CCSDT or something. But what we are doing is we are at an atomistic level, which is the yellow level there. People can typically now go to 1 to 10 microseconds, but that's essentially not sufficient for seeing things that we want to see. If you think of a nitrate transport process, when the nitrate transporter is not phosphorylated, the actual real time it takes for a nitrate to transport would be hundreds of microseconds. So we have something which is state of the art is roughly 10 microseconds, or that's what you're going to see in very, very high impact papers coming out of molecular simulation field. And the reason for that is this, that if I have a laptop and I'm trying to do these simulations, and you can see from this thing at the bottom, that on a simple laptop, a microsecond takes a year. So if I'm thinking about a millisecond, it's 1,000 years. That's 200 graduate students' lifetime. So that's it. But we have one big way is a brute force approach is that we have this huge computer, and you're sitting in the building where this this is not right here, but this is the place which got this computer built, NCSA. So Blue Water supercomputer on our campus, we can actually utilize it to do these type of large calculations. But again, even that is limiting, and we'll talk about how we can go beyond even Blue Waters. So this is an example of a work that was done on Blue Waters. That's not from me, but from the theoretical computational biophysics group. That was Professor Schulten was supposed to be here, but unfortunately not. So he has done this model of an entire viral capsid, where every single atom on the viral capsid is in this atomistic detail, and he's built the structure of this entire viral capsid. Millions and millions of atoms in this. But obviously, he's not doing dynamics on these things. It's just a snapshot of a viral capsid that has been built. And it was done right here at Illinois. This was on the cover of the Nature in 2015. But imagine now that, let's say I have built the world's biggest computer, and I can run all these things for like a very, very long time. But even then, I end up with tons and tons of terabytes and terabytes of data. How do I make sense of all of that data, right? So you have these simulations, which are 100,000 atoms, and you're saving those positions of those 100,000 atoms every femtosecond. And so imagine how big these data sets tend to be. They're running in hundreds of terabytes. So I cannot even transfer the data from the computer to my laptop to analyze it. Analysis also has to be done on a supercomputer when you're doing these type of very large simulations. So, and then the question obviously becomes that if I have so much of data, how do I make sense out of it, or how do I convert this information into knowledge? What we do is one way which we are using is we're trying to make Markov state models of these proteins. And I think all of you, everybody here would remember doing a small chemical kinetics problem where A goes to B and you have two rates connecting them. But you can actually have a slightly complicated network. You can have A, B, and C, D, and then now you have 16 rates connecting them. And if I know all the rates of transitions between A, B, and C, D, I can actually write down a very, very simple chemical kinetics equation, and I can get the population of these states. Now think of these states in a chemical reaction as conformations of the protein. One is an outward open, one is an inward open, one is occluded, or something else. And if you can just calculate all the rates of transition between these different conformations, I can put together the dynamics of this protein as if I was observing it in a real cell, if my model is accurate at an atomistic level. So, and that's what we try to do. We can also use this type of a formulation for sampling. For example, what we do is you can start from the only known structure that I have, that is an inward open structure of this particular receptor. And then we're using, let's say, 100,000 machines at a time, and we are running these simulations. And then we discover some new or slightly different structures from what we already had. And then you can, so this is basically a graph search algorithm for those who are in computer science. So you're basically searching through, trying to find out all the nodes in this graph and try traversing all the edges. And you can think of a lot of algorithms that can be used here to sample this. So you can keep on going, and you found occluded states, you can find an outward open state. So we do this a lot of times, almost every day. So there's a data that comes every day. You cluster that data, find out new states, you start again, and then there's an automatic way of generating these landscape. Now, and then once you have this data, you cluster it into a lot of states. You find out what are the rate of transition between these different conformations. If the transitions are really, really fast, you can club them together into bigger states, which make more sense to a non-expert. And now we can build this type of a network model for the protein dynamics, which gives you the accurate description of the dynamics. And now we can take this type of a network and start predicting experiments. I can start predicting NMR. I can start predicting DR spectroscopy. I can start predicting HDX. Any experimental technique that we think of, which could be calculated based on the positions of the atoms in a protein as a function of time. So what we have done is, there are a lot of, I'm not going to talk about that work, but in past work we've done a lot of comparisons with accurate experimental data. We successfully match all sorts of experiments from this very ab initio model. Now, I'll give you an example of this type of a sampling and stuff on phosphorylated and an unphosphorylated transporter. So we have this single dot here on this landscape, and the bars here or colors represent the density, red being the low density, or free energy, so red being or white being low. So we started from a single structure. Let's say we are sampling this landscape for both phosphorylated transporter and an unphosphorylated transporter. And we can keep on doing this sampling, and we are exploring more and more space until we reach this sort of a converged landscape of description. So here what you're seeing is the top distance and the bottom distance of the transporter, and that can characterize this type of a transition. So you can see an occluded state, an inward open state, and outward open state by just looking at these two metrics here. Now we can start asking scientific questions like, why is a phosphorylated transporter better than an unphosphorylated transporter for transporting nitrate? What you find is that the unphosphorylated transporter never really goes to an outward open state. So if it's not in an outward open state, it can never catch a nitrate. So it's a very, very high energy state for an unphosphorylated transporter to go to an outward open. So it never catches it's a very rare event for it to catch a nitrate, therefore the transport rates are very, really low. So again, we can start asking a lot of scientific questions once we have the model. We can start asking about the effects of individual mutations that are going to happen on a protein and the transport rate. And now we have a prediction, which is we have a single crystal structure. Now we have predictions for all the structures, not just these two. These are the two main that we have shown you. And you can see how the tunnels are logged at one in the bottom and others. And then you can obviously also start asking questions about what are the transition pathways. How do I go from one structure to the other? What kind of changes have to happen in the protein if I want to go from one structure to the other? And we can draw this type of network graph, and I can show you which individual structures I have visited on en route during the nitrate transport cycle. So this information is really, really useful. It definitely complements what we can learn from crystallography, from NMR. And this is a quick movie of a transport. It's not a very high quality, not spend much time on it. But you're seeing a nitrate coming from outside into the transporter, and there are gates which sort of obstructs this path. There are charged residues that hold it into the middle. Protein comes into an occluded state and sort of squeezes it inside. So this is a slow process. This is an intermediate state which is really stable. Nitrate is never really going out. It has to break all these charged. What happened to it? I think it's my presentation was like 80 MB, so there's some issues. But anyways, you see this type of a transport process, and then we can characterize it. Not just one mode of. I showed you one particular, a reconstruction of this type of a transport, but you can do a lot of them. Similarly, you can also start looking at other problems using molecular models. We work a lot with plant hormones. I'll show you just one example of that. And what we are trying to do with these hormones is we're trying to understand first how these hormones are perceived in the first place. So we have crystal structure for some of the receptors that recognize these particular hormones in plants. We're saying how would it recognize a receptor. So this is an example of again, I'm showing you a free energy landscape. Red is like low populated states. Blues are highly populated state. And what we're seeing is a pathway for this type of a ligand. So this is for example, this is a simulation of a drug which is trying to bind to this particular receptor, and it's a very popular cancer drug. It burns billions of dollars for Novartis. And we don't actually know why this drug works. And now we are able to simulate these things in detail to see these type of processes. It's taking time. Similar thing with plant hormones. We can also simulate these plant hormones and see how they are perceived. There is still receptors that have not been crystallized for plant hormones, like ethylene. We don't have anything for that. Even for some of the receptors for which we have structures, like auxin, there are only one or two structures of that receptor. So these type of methods could augment our understanding of these plant hormones. And what you're seeing here is a sort of a simulation we did for the ABA receptor, which is an ABA is a drought resistance related hormone, and how it is recognized by its receptor. The value of these simulations comes in basically identifying the intermediate states and the barriers to transition. So this hormone can actually diffuse and bind this receptor, but there are huge barriers to it for it to come inside the pocket. So you're seeing there is almost a region around 5 on the x-axis where there is absolutely very little probability or free energy is really, really high. So there's a huge transition for this particular hormone to bind to a lysine, a conserved lysine in the pocket. And that is the key barrier. And now we can say, what are the key mutations I can make that would reduce that barrier? So you can have a hormone at a low concentration, but it can bind the receptor, and then it can do the function. So these are, again, first principle predictions we can make for these type of systems. I also worked with Steve Huber in plant biology on brassinosteroid signaling. We are trying to understand how these particular receptors identify this particular growth hormone in plants. And I'll be quick. We work with particular kinases that are involved in those pathways. This is a protein that signals plants to grow, and it's also in humans. This is a protein that signals our cells to grow. So if they're not working, you get cancer properly. And I've done a lot of work on these particular type of enzymes. We did a lot of simulations of these receptors, and what we found is that there's a particular mechanism by which they regulate their activity, and that is an unfolding of a helix. So you see this orange helix there, and that actually unfolds with a high RMSD, and then we get an inactive form of this enzyme. So only 5% of the time this enzyme is really active based on our predictions. Can we make it more active? We have tried some strategies. We are still testing them experimentally. We were trying to put a more stable helix and engineer this protein to be more active so that more growth could be promoted and stuff like that. And finally, so we do all these things with plants, with plant hormones, with nitrate transporters. We are also looking now at Rubisco a little bit. But we did all this work that is done on Blue Water supercomputer, but we can also do a larger scale distributed computing where we can distribute these individual simulations on a graph to a lot of volunteers. So I work with Folding at Home at Stanford University. It's a consortium. It's a group of scientists who have come together, and roughly 1.6 million people contribute computer time to that project. So at a given time, you could install that on your computer, and your computer is not doing any work. It's plugged to the power. We can run simulations on your machines, and that data comes back to us, and we analyze it, send you new simulations to run. And in that way, we've been able to build some of the world's largest molecular simulation data sets. One example of that would be, I know that a couple of people from Google are here. There's some work I did in 2014 with Google. I've worked with Brian Kobilka at Stanford University on simulating this particular type of receptors called G protein coupled receptors. We're trying to understand how these receptors work. These are the receptors that bind caffeine molecule. These are the receptors that bind almost half of all the drugs that people take. They would actually go in and bind to these receptors, and they activate processes in our body that are responsible for a lot of diseases, for curing a lot of diseases. So we understood how this receptor works. There are different parts of the protein that need to change their shape, and therefore then the receptor gets activated. And we used Google Exacycle for that. And Google Exacycle is a platform where we're doing all these simulations on Google's resources when you're not doing search. So it's like roughly several million processors where we can use them for a few hours every day, and you can do a very large simulation. It was on the cover of Nature Chemistry, and this was a collaboration with, as I said, Google. There are a couple of Google scientists who helped us set up this type of a platform. And finally, I will just have a few comments on what we want to do for molecular models and integrating it with higher levels. So there are two approaches that are basically being taken. The first approach is what, and both these approaches are happening at Illinois. Obviously, this work is from Klaus Schulten, and this is the entire chromatophore modeled in atomistic detail. So again, millions and millions of atoms, and you have a clear description of what this machinery is doing in nature. What I'm doing, though, individually is that we are looking at individual pathways. I mean, this is an example of a brassinosteroid pathway. It could be any other pathway that we could work on. We're trying to understand the individual pathways by breaking down it into smaller and smaller components and understanding interactions of the individual proteins in the pathway. So you have these network models for for cellular signaling. What we are doing is we are opening that box, and we are saying what is inside that box, how can I regulate that box? So this is an approach that I'm taking. There's also a whole cell approach where people are doing first principles entire cell now. And that's going to be a huge project whenever the new supercomputer comes to Illinois in 2020 at some point. So what is the other thing? So one other thing that you have found that is like really that we have found synergistic in this work and with all the work that has been presented in the conference is that network models are the natural framework for integrating things with your metabolic models, with any genomic or gene network models. So Markov state models are essentially have the same network, so you need the same type of standards. So you can have a gene network model in SBML. You can also put in my protein conformational network models in SBML. So there could be a lot of interoperability if you're going from a molecular to a higher level model. And traditional approach to molecular modeling has been that you look at a single observable, which is some property of that protein, whether it is folded or it is not folded, whether it is functioning or it's not functioning, and you measure it as a function of a free energy, and you get some understanding of what is happening, but behind all those free energy plots that people talk about in molecular modeling, there's an underlying network of conformation which is not visible. So I think what our work is doing is making, and same thing I could say for all the models that we have seen for photosynthesis. We have proteins which are boxes which are interacting and with arrows between them. But what we don't see are how these individual boxes are being regulated, and can we integrate that information in a useful way, or can we ask mechanistic questions going downwards. Same thing, one other final comment I have about this type of work that we are doing is that I think computational structural biology could be a really, really big thing for plant biology. Because really we do, the biology is not that, in terms of the tools that are available to people to do plant biology are limited as far as I can see coming from medicine. And there is very limited amount of structural work that is happening in comparison to medicine. So what computational structural biology can do for plants is that we can help you see all these processes. Even if you don't have a structure of a protein, we can have a guess a structure and validate it using coarse experimental information. So we can really provide, and that's a very, very important message from my talk here. If you look at a protein database, 100,000 structures, it has been 100 years of X-ray crystallography. We got 100,000 structures for medicine purposes. For plant biology, we are at few thousand, or even less, for key proteins. So we need to really, I think this is something that can augment the research in the plant field. And I think I'll have a lot of people to thank, Professor Steve Long, Amy, for organizing this session, for being part of the SCI, involving me in these activities. Professor Steve Huber, I talked very briefly of his work, but I work very closely with him. And that's my group. It's a little pixelated, but these are like 5 graduate students and 1 postdoc. We work, I'm a Blue Waters professor, so we use a lot of computing time on Blue Waters. I've done some work with Google Exacycle, with Amazon Web Services. We have a huge grant from them to run simulations on Amazon Web Services now. And as I said, I'm part of the Folding at Home where Vijay Pandey is the PI. So we're doing a lot of distributed computing with all these resources. So with that, I'll thank you for your time.